Good afternoon, everyone. I am Fauzia from Department of Biotechnology Graduate School, Universitas Gadjah Mada, Yogyakarta. Proudly present our abstract, Cytotoxic Analysis of Hesperidin and PGV1 for Hep-G2 Hepatocellular Carcinoma Cell by Bioinformatics and In-Vitro Study. Together with Ms. Febri Wulandari, Dr. Muti Ikawati, and Professor Edi Meyanto. Hepatocellular carcinoma is the most common type of primary liver cancer in adults and is currently the most common cause of death in people with cirrhosis. Hepatocellular carcinoma is the third leading cause of cancer-related death worldwide. Cancer treatment can be broadly divided into localized and systemic treatment. Cytotoxic chemotherapy is the systemic treatment. It means that in the theory, it's whole body usually via the bloodstream. Although in the practice, the ability to treat the whole body depends on its ability to get into all tissue. This is in contrast to surgery and radiotherapy, which treatment localized area. Other systemic include hormone and newer targeted therapy and immunotherapy. So far, treatment with chemotherapy has shown unsatisfactory effect and low cell selectivity, so that the normal cells are negatively affected by drugs such as doxorubicin. This necessitates the development of chemotherapeutic agent with low side effect and high selectivity. The Faculty of Pharmacy UGM has developed pentagamma-funon or PGV1, which is a monocarbonyl analog of curcumin. PGV1 is known to have promising activity as a chemotherapeutic agent on various cancer cell lines. Inhibitor of the cell proliferation can occur in the presence of the cell cycle arrest, both GO, G1, S, G2, and mitotic phase. PGV1 is known to inhibit cell cycle in G2M. Hesperidin is a flavonoid that is commonly found in orange peel. Hesperidin is known to inhibit cell cycle, induce senescence, apoptosis, stimulate oxidative stress in cancer cell. This study aimed to predict the protein target of hesperidin and PGV1 by using bioinformatic approach and to determine the cytotoxic effect of both compound of Hep-G2 hepatocellular carcinoma cell. And then targeted the bioinformatics study, which carried on CREPET, PubChem, Swiss target prediction, GenCard, Vulkan, and interactive fan. And the in-vitro study observed using the direct counting method. The result of the target protein screening PGV1 and hesperidin are then sliced with protein are overexpressed by hepatocellular carcinoma using the diagram fan. Figure 1 shows that the 100 prediction protein targeted by using hesperidin and PGV1. There are 3 proteins are overexpressed by hepatocellular carcinoma. EPHX2, ELB, and F2. So these two compounds show the ability target protein that are overexpressed by hepatocellular carcinoma. Once know the predict protein are targeted using Swiss target prediction. Further exploration of the expression of these 3 protein target was conducted on the TCGA data set through the Vulkan website. Figure Figure A show that the expression of EPX2 protein. Figure B, similarly to OLB protein, and figure C is expression of the protein F2. The high expression of all three target protein in both tumor cell when compared to normal cell suggests that these protein have an important role in modulating cell cycle in hepatocellular carcinoma. To find out more about the effect of highly expression of these three protein in patients with hepatocellular carcinoma, further search were conducted to see the survival rates of patients who overexpress the three protein. Figure D show the survival of sample with high protein expression in red line and low protein expression on blue line. The results show that overexpression of all these Hesperidin and PGV1 protein target had advanced effect on the survival of hepatocellular carcinoma. So these three targets become important target in hepatocellular carcinoma. The PGV1 curcumyl analog and Hesperidin compounds show that the ability to inhibit proliferation and Hep-G2 cancer cell with cytotoxic activity. The cytotoxic activity of PGV1 was greater than that of Hesperidin, with IC50 value of PGV1 of 0.86 micromolar and Hesperidin of 105.39 micromolar. The highest cytotoxic activity of PGV1 is in line with previous study on previous cancer cell line. In conclusion, it can be predicted that the ability of Hesperidin and PGV1 to modulate the cell cycle that causes Hep-G2 cancer cell death in relate to the inhibition of this protein. In addition, IC50 value can be used for advanced cytotoxic analysis, that is combination cytotoxic analysis. This is the reference, and thank you very much for your attention. See you.